All right, so this is a video to serve as a makeup video for those of you who might miss the lab. So I'm just going to go through what we did in the lab so that way you can fill out the lab handout and have that as a resource for the quiz. So this was our Osmosis Lab. There were a couple materials we used in the lab, but the main two materials would have been these solutions you can see here and a dialysis tube bag. So these are the solutions that we used. You can see they're labeled by their concentration of solute in each solution. And you can see the one with the highest concentration would be this blue bottle here, where it's 1 molar of that solute, and the smallest concentration would be this one here, 0.2. So those numbers represent how much solute concentration there was in each solution. So each group was assigned 2 different solutions. One was going to go on the interior of their dialysis bag, and the other was going to go on the exterior solution. That dialysis bag, you'll see in a minute, serves as a semi-permeable membrane, similar to the cell membrane, where it's allowing water to pass through, but blocking the solute's passage through the membrane. So this video shows the initial setup of our bag. So you'll see initially here, there's a blue dialysis bag. It has the 1 molar solute concentration, and the exterior solution is going to be the 0.2 molar concentration. So from this setup, you should be able to determine which solution is going to be hypertonic to the other and form a hypothesis. So the hypothesis students were making was basically focusing on, again, the tonicities of the solutions inside and outside the bag and their prediction on where water was going to go based on those tonicities. So you can go ahead and fill out the hypothesis section on the second page of your lab handout before proceeding to the actual lab execution. So I would pause the video here and write down your hypothesis using that if-then format and those vocabulary terms of hypertonic, hypotonic, if water is going to exit the dialysis bag or enter the dialysis bag. And you can also use the terms gaining mass or losing mass, because that's how we are going to collect our data. So I would, again, pause here, write your hypothesis. Okay, so this next video shows me taking the initial mass of the dialysis bag. So there's going to be 2 masses shown here. The one that I'm showing right now is the initial, where the bag was massed before being submerged in that green solution from the last video. So you'll be able to see the mass and record this in your data table. So you'll be able to see the mass was about 15.04, 15.05 grams before being submerged in that bag. So go ahead and write 15.04 or 15.05 grams in your data table on that first box. So after taking the mass, groups went back to their tables, and all they did was put their dialysis bag in the exterior solution and wait for 25 minutes for the osmosis to actually occur. So they set this up, and then they proceeded to work on their pre-lab questions from the first page of that handout. After those 25 minutes were done, the same procedure of taking the mass of the bag was repeated, where the groups went back to this balance, and they were able to collect their final data. So in this case, you'll be able to see the new mass of our bag is 16.97 grams. So you can record that as the second box in that data table. Okay, so that's basically what we did in the lab. It was pretty straightforward, not a lot of moving parts. So just based on this part of the video, you should be able to answer all of those lab questions under the data section and the first conclusion question. The final conclusion question, all students were given data from different trials of the experiment, so different interior and exterior concentration solutions, and they were able to see some data that differed from theirs because it was a different, again, combination of solutions. So for that final conclusion question, you will use this data, pick one of the trials that is different from ours. So any of them, but this section right here in the middle, you can pick and compare it to the trial you just saw in this video. So like I just mentioned, you should be able to fill out the entirety of that lab handout now that you saw how the lab was conducted, and this lab handout is able to be used on our quiz this week. So make sure you have your questions filled out and the data section and conclusion section filled out so you can fully use this resource on the quiz. I hope this helps. Let me know if you have any questions.